guys, welcome to the Road to Success MCAT Test Prep series. My name is AJ, and this video is the first in a new series on my channel where I'll be discussing everything you need to know about the psychology section of the MCAT. Starting off, we will be talking about the subject that is sensory perception. Sensory perception consists of sensation thresholds, sensory adaptation, Gestalt laws, and vision. Vision as well as hearing will also be the most tested senses on the exam, so they will be taking up the bulk of this video. That being said, somatosensation, olfaction, and gustation will be covered as minor subtopics at the very end. We have a lot to cover today, so let's jump right into it.
Alright, I'll start off with sensation thresholds. Sensation thresholds are basically our body's way of gatekeeping perception. Otherwise, we will constantly be overloaded with stimuli. The absolute threshold of stimulation is the minimum threshold of stimulus that we need to detect the stimulus 50% of the time. The subliminal stimuli is everything that we don't, or that doesn't reach that threshold. We will not be detecting these. The just noticeable difference, or the J&D, is the minimum difference that can be detected 50% of the time. Basically, the absolute threshold is the minimum amount, while the just noticeable difference is the difference in intensity added by an additional stimulus. Now, let's talk about Weber's law. Weber's law is an application of the J&D that describes the minimum difference as a ratio. So, for example, if you hold a 2-pound weight and you increase it to 2.5 pounds, you will notice a difference. But the same can't be said if you hold a 20-pound weight and add the extra half pound. It must be a proportion. Weber's law states that a change in intensity, or delta I, over its initial intensity, which is regular I, equals a constant K. So for my previous example, 2.5 pounds minus the initial 2.2 pounds over, like I said, the initial 2 pounds will equal 2.5 minus 2, 0.5, or sorry, I was jumping the gun there, I was doing the math too fast. It's 0.5 over 2, which will be 0.25, which will be your K value. Now, these are the main senses that we have, and this is how we adapt to them. Feel free to pause and take notes, as these are essentially all that you need to know for what the adaptations are. For the MCAT, though, the most important senses are sight and hearing, which will be covered in depth in this video. Before we jump into those, let's talk about the basics of perception. Starting with processing types. Bottom-up processing is when the stimulus influences our perception. We take in our surroundings and we use what we observe to reach a conclusion. This is why bottom-up processing is considered to be more accurate. Meanwhile, top-down processing is sort of the opposite. Our previous knowledge influences our perception. The schemas and memories in our brain are used to interpret the stimulus observed and to reach a conclusion. An easy way to differentiate between these types of processing is by remembering that bottom-up is specific to general, while top-down is general to specific. Now let's talk about Gestalts. Our Gestalts are basically our brain's way of processing how the whole is bigger than the parts. We want to make sense of the seemingly random, and we sure as hell will make sense of it. Do not worry about the principles of enclosure, connection, I'll make this a bit bigger, yeah, and figure to ground. The MCAT won't be testing these, but they will be focusing on the rest. So, starting with the law of proximity, it states that the objects close together will be grouped together. The law of similarity states that similar objects will be perceived together. The law of symmetry states that objects with lines of symmetry will be associated with each other. The law of closure states that objects grouped together with distinct openings will be associated as a whole, as you can see here with the arc and the lines. This looks like a rectangle, this looks like a circle. The law of continuity states that we see lines as following the smoothest path. Now, one last law that isn't on this infographic is the law of pragnans. The law of pragnans states that reality is often organized and reduced to the simplest form possible. So, for example, let me draw 5 rings right here. It's a bit rough, but bear with me for a second. Instead of seeing these 5 overlapping rings, Your brain, assuming you've seen the logo, you will be perceiving these as Olympic rings. One concept instead of 5 random objects. So now let's move on to vision. The anatomy of the eye can be studied on your own time, but I will mainly be focusing on rods and cones for now. These are our photoreceptors, and they perceive light and color. Starting with rods, they are our light perceptors and they are used for light and night vision. They're located in the periphery areas of our eyes and they are extremely sensitive and will take a long time to recover. This is why we need some time to adjust our vision whenever we turn off our lights. Cones, on the other hand, these are used for color vision. They're located in the fovea, which is near the macula, so somewhere here. They are much less sensitive than rods. Now, one thing that they both have in common, though, is that they both perceive light through the phototransduction cascade. I know this looks really complex, but bear with me. We just need to know the 6 steps. So, when light hits rhodopsin, the cisretinal inside of it will change into transretinal, therefore changing the conformation of rhodopsin. This will cause transducin. Where is transducin? I guess it's not drawn on here. Oh, it is here. I'm so sorry. This will cause transducin to break off from rhodopsin and activate phosphodiesterase, or PDE. PDE will convert cGMP to GMP. So from cGMP to GMP, there will be less cGMP, causing sodium channels to close, leading to hyperpolarization of the cell. When that occurs, the rod or cone will turn off and the bipolar cell will turn on. This process is the same in cones, like I just said before, but instead of using rhodopsin, they'll be using photopsin. Now, lastly, let's talk about visual field processing. Take notes on this diagram as it covers basically everything you need to know. I'll wrap this up by talking about some miscellaneous vision topics. Magnocellular pathways and parvocellular pathways are basically buzzwords that you have to associate with motion and shape, respectively. The actual anatomy of these pathways are not important for the MCAT, so don't worry about that. Trichromatic theory states that we see the world in combinations of with colors red, green, and blue, your primary colors. Now lastly, parallel processing states that we perceive several stimuli at the same time. Now let's move on to sound. Like with the eye, the anatomy of the ear can be studied on your own time, but I recommend you look into the functions of the malleus, the incus, and the stapes. These three objects right here. The semicircular canals I'll also recommend. On the right, we have the cochlea. So right here, we have the cochlea. And this is where the fluid is pushed to stimulate sound perception at the organ of Corti. Sound processing occurs by activating the hair cells of the organ of Corti. The hair cells, or kinocilia, are activated by exposure to potassium, which is pushed in by endolymph. This will cause an active potential to be sent to the auditory nerve. Keep in mind that the location of the hair cells in the cochlea play a role in how sound is perceived. Hair cells at the base are perceived as high-pitched, while hair cells at the apex are perceived as low-pitched. This is called basilar tuning. Now let's move on to somatosensation. The types of somatosensation are pressure, temperature, pain, and position. Each part of our body is perceived in a specific part of our brain, as you can see on the right in this diagram right here. Note that our hands, feet, and face have a lot more nerves than the other parts of our body. That's why it hurts so much when you stub your toe. Positioning, or proprioception, is a cognitive process where your brain recognizes that you are in space and positions itself in an appropriate way. Kinesthesia is a similar process, but it's more behavioral. It is how our body moves. Lastly, let's talk about pain and temperature. For pain, recognize the TRPV1 receptor. It's what allows us to detect heat. As for pain, it is processed by various different receptors. They are all on the right, and please note that C fibers are for lingering pain, as they are very slow, because they are not myelinated. Meanwhile, alpha delta fibers, they are myelinated, so they tend to be a lot more fast, leading to sharper pain. Alright, olfaction. There isn't much to cover here apart from anatomy, but please note that smell enters in from your nose, and it'll enter and it'll reach the olfactory bulb via the passing through the olfactory epithelium here, entering past the cribriform plate, reaching the olfactory bulb, and eventually traveling up through the thalamus. I wrote here, note the existence of pheromones. This is an animal concept, and the main thing I wanted to talk about it really is that it causes innate responses in animals, but they do nothing to us, and as a result, they're not used by us. So don't associate pheromones with humans, they're solely for animals. And now finally, we reached gustation. There really isn't much to discuss here either, since it's very low yield, but one thing that I want you guys to know is that sour and salty taste buds rely on ion channels. Meanwhile, bitter and umami taste, they rely on G protein coupled receptors, so I'm going to write GPCR. So yeah, that's basically all I have for you guys. That's all I have for sensory perception. There's a lot of stuff that we covered here. I want you guys mainly to focus on vision, sound, and if there's any equation that you have to memorize here, it would definitely be Weber's law. Apart from that, that's pretty much covers everything I want to cover you guys about sensory perception. I hope you guys learned something. Have a great rest of your day, and happy studying.